Hello, everyone. I'm Tim Hanks, and today I'm going to tell you about collaborative research that's being conducted between my own lab and that of Rishi Chowdhury, both of us at the University of California, Davis at the Center for Neuroscience. Now, this is research that was very recently supported by the CRCNS program, but it builds on a foundation of research that both of us have conducted in the domain of decision making, and combines it with newer techniques that we have to measure activity of hundreds of neurons simultaneously. And so we hope that the ability to record those neurons simultaneously will give us insights in order to develop multi-region network models of cortical and subcortical interactions for dynamical control of decision making. Now, as my title already gives away, we are going to be looking in this talk at one particular task that involves dynamical control of decision-making. That's a change point detection decision-making task. This task we call the Poisson-Clicks change detection task. The way it works is that rats are placed in a behavioral box that has two nose ports. They're cued with a light to poke their nose into one of those ports, and then they're presented with an auditory stimulus. And this stimulus consists of a sequence of auditory clicks that are generated by a random Poisson process. Initially, the underlying rate of that process is relatively low, but then at a random, unpredictable time on each trial that varies from trial to trial, that Poisson rate increases. And the rat's task is to detect and report when that increase occurs. Now, we give the rats a limited response window to report these changes, typically less than 1 second. If they don't report the change in time, that's an incorrect trial, that's a miss. If they report the change too early, that's also an incorrect trial, that's a false alarm. And if they report the change during the response window, they can go and get a reward. Now, rats can do this task reasonably well. This is a psychometric function showing their hit rate as a function of the change magnitude. And so when the change magnitude is very large, they get almost all of the trials correct. They are able to detect all of those changes, and their performance diminishes as you would expect when the change magnitude is smaller. We can also look at the animal's reaction times, which have an orderly relationship with change magnitude as well. The animals respond more quickly when there's a large magnitude change that's easy to detect, and they take more time when there's a smaller magnitude change that's harder to detect. And all of these behavioral results are consistent with the dominant models of decision making in the field, which I don't have time to tell you about here today because instead I want to focus on what we consider to be one of the central leverage points of the new experiments that we've been conducting. And that central leverage point is the ability to do high-density neural recordings with neural pixels probes. And so many of you are probably familiar with these probes, but the basic way in which they work is that they have densely packed recording sites, hundreds of them, along the recording shank. The recording shanks are long enough that we can penetrate to get both cortical and subcortical regions on the same probe. This is the setup that we use in the lab, and so we can record from multiple sites across the brain in awake behaving rats while they're engaged in those decision-making tasks. And we use this to record from a network of brain regions that have been implicated to be involved in decision-making, including those illustrated here, the posterior parietal cortex, which I've labeled PPC, the frontal orienting field, or FOF. Both of those are cortical regions, and also the subcortical regions of the anterior dorsal striatum, labeled here ADS, and the superior colliculus, or SC. Here's a geometric construction of the way in which we do this with two probes. Again, each of these probes individually can get two regions. So with one of them, we can get both PPC and the superior colliculus, and the other one, we can get FOF and the striatum. This works pretty well. Here's an example of recordings from a single probe that's targeted at an angle to hit both the frontal orienting field and cortex and the underlying anterior dorsal striatum. And with these probes, we can typically record from many dozens to over 100 neurons per brain region per individual session. And in addition to that, we can get just as many or oftentimes more multi-unit clusters. And just to really put that in perspective, it really has been a game changer for us. I tabulated the numbers, and the first year that we did NeuroPixels recordings in my lab, we had recorded more units than I had in the entirety of my career up to that point. In addition to that, and I would argue perhaps even more importantly, it gives us this snapshot of population level activity at each moment in time. And that allows us to do single trial analyses that were not achievable with the methods that we had previously used. And I'll come back to that in just a moment. Before I do so, though, let me just tell you a little bit about the basic response properties that we can measure in this task. And for the rest of the talk, I'm just going to focus on FOF and the anterior dorsal striatum, and for the next slide, only the anterior dorsal striatum. So one of the first questions that we've addressed is whether the neural responses that we're measuring reflect the evidence evaluation process that goes into the animal's decisions. And so what I'm showing you here are PSTHs from neurons in ADS that do reflect that. And I've aligned these responses to the time of the change in the auditory stimulus. This is the increase in that Poisson rate that the animal has to detect. Now, I've also sorted the neural responses based on the magnitude of the change in the stimulus. And what you can see is that when there's a larger change in this auditory stimulus, it's easier to detect. We also have a correspondingly larger change in the neural responses, and that change occurs earlier, which is also consistent with the animal's reaction times. And so this tells us that these responses really are reflecting evidence accumulation and reflecting something related to the decision process. Now, this is all well and good, and there are many different analyses that we have been performing and can continue to perform along these lines, but today I really want to focus in on preliminary analyses that we've been doing that look at the population level activity rather than considering one neuron at a time. And so one of the first things we can do to look at population activity is just to look at how this activity unfolds over time during different epochs of the task. And we can split the task into two different epochs. The decision interval, this is the time period where the animal is stationary, listening to the clicks, but is not yet reported its choice, so it's deliberating, but is not yet decided. And the movement interval, when they're reporting a choice and going to get a reward. Now, all of these analyses are done in one single session individually because all the neurons are recorded at the same time. And I'm going to break this down into the two different brain regions that we targeted with this probe. In the frontal orienting field, we had 111 units in this session. We looked at the population trajectory during this decision interval projected onto the first two principal components. And you can see that it moves through this principal component space. Now, one of the first questions we want to address is whether this is consistent across a session. So I should mention that for each of these traces, it's an average of 10 consecutive trials, so it's chunks of trials. And if we look at the other chunks of trials within the same session, we can see that there's a decent amount of consistency, generally moving from the bottom right over towards the top left in this principal component space. We can also look at the trajectories during the movement interval, and these are from the same population of neurons. And even looking at just the first chunk of trials, you can already see that there's some differences in the dynamics in which they have more of a rotational characteristic. And this is also consistent across different chunks within the same session. Now, this is for the frontal orienting field. Like I mentioned, at the same time on the same probe, same animal, same session, we're also recording units in the anterior dorsal striatum, an underlying structure that's densely innervated by the frontal orienting field. And looking at the decision interval, it's actually a bit more complex than in the frontal orienting field. There's actually a bit of an out and back movement within this PC space before moving off to the side, but not much in the way of rotational characteristics. If we look at the movement interval in the ADS, we have at least a hint of rotational dynamics here as well. Now it's moving in this space in a counterclockwise rather than clockwise direction. But nonetheless, we also see a general consistency across different chunks of trials within these sessions. Now, given this consistency of the trajectories across a session, one of the next questions that we wanted to address was simply, can we decode time from these neural responses? And to explain how we do this, there's a lot of other factors that are happening, events that are happening during this task, and we wanted to divorce this from those confounds. So we focused in on this variable wait time interval. This is what I was calling the decision interval before. The animal is stationary, listening to the clicks, but has not yet made a choice, and the click rate has not yet changed. And we looked at the first 800 milliseconds of this interval. We broke it up into 32 different time bins, and we simply asked, can we predict or classify the time bin based on the neural response alone? And so to build up how we did this, here I'm looking at the 7th time bin. So this is a time bin that starts 150 milliseconds after the beginning of the trial. And what we are doing is leave one trial out, decoding. So we take all the other trials, build our classifier, apply it to the firing rate in this trial at different time points, and then try to predict what the time point is. Now, for these analyses, there were around 250 trials that were long enough to go into the analyses. There's 32 time bins. So chance performance here would be 1 out of 32, which would be around 3.2%. We got the correct time bin 30 times, which is 12%. So that's a relatively low, modest level if you just look at it in absolute numbers. But if you also look at the misclassifications, it's informing as well. Because most of these misclassifications are being classified as adjacent time bins or nearly adjacent. Almost all of them are within 100 milliseconds. Keep in mind here that these time bins are very narrow, only 25 milliseconds in length. And so if we build out the full confusion matrix, not just the 7th time bin, but all of the time bins, where the true time bin is along the y-axis, the predicted time bin from the classifiers along the x-axis, you can see that the density is very heavily concentrated along the diagonal, which would be perfect classification, or very close to the diagonal. There's very few misclassifications that are far from the diagonal. Now, like the previous talk, I don't know exactly what I should have expected going into this, but I was surprised at the level of precision and robustness at which we could decode time from these responses. And I think just the key thing there is that we're recording so many neurons at the same time. And I think that's an important message here is how much you gain from being able to make these large-scale population-level measurements. And so just with the last couple minutes I have, I want to give you a sense of where we're taking this. We believe that our results so far are consistent with this emerging picture that's come from other labs, suggesting that activity in networks that are related to decision-making unfolds along a consistent manifold during decision processes, and we believe that's why we can decode time so well. And given that we have recordings from multiple brain regions, we're interested and continuing to ask questions about how these dynamics are controlled and or shaped by interareal interactions, and what are the mechanisms that allow this robustness of neural dynamics. We find that the responses are precise to the level of tens of milliseconds, so what are the key ingredients needed in networks to allow that to occur? And finally, with any remaining time before taking questions, I want to acknowledge the people I have the good fortune to work with on a daily basis, and especially recognize 3 individuals: Adam Goldring, who is the postdoc in my lab, who was the lead experimentalist collecting all of these data; Tanner Stevenson, a shared grad student between my lab and Rishi's lab, who did lots of bridging work on this; and Ananditha Day, a graduate student in Rishi's lab, who is moving on to a postdoc at NYU, who did all of this state space analysis. So thank you very much.
Questions? Yes, please, just a moment.
I can finish out. It's
Yeah.
a really neat task and everything. I was curious how you thought, is it important for there to be a functional purpose to record time in this task, and whether that, might affect the result? I can imagine the longer you wait, the more likely it is that there's a change.
Yeah, exactly. So in this case, the particular point you made is not true because we have a flat hazard function, so it's exponential distribution. But the more general question is, what is the purpose of time here? And so we don't necessarily think it's time per se. We think it might be time that's used in an important way for the decision process. So even though there's not an increasing probability of a change as time elapses, there still is behavioral urgency. So the animals are more likely to report a change. They have a behavior that's consistent with a non-flat hazard. So perhaps you're seeing something that's more related to urgency to decide, from which you can decode time. So we're not quite sure what those signals are being used for, but they relate in a very precise way to time as it unfolds in the task.
Very exciting data. So 2 questions. One is, have you looked at eye movements? That's one thing I'm sort of wondering, because you look at regions which are highly involved in eye movements, right? And the second question is, did you try to decode the clicks themselves, given the richness of your data?
Right. So first on eye movements, these are regions, we call them more orienting regions for the rat, because they do have eye movements, but the bigger way in which they change orienting is with full body motion. They are stationary in the port during this time. I say stationary, they have to obstruct the laser, but they can still fidget around. So others have found that these fidgets really matter, and Churchland's lab has shown a lot of work on this, and that's something that we need to look at in more detail. And then your second question was related to can we decode the clicks? And there's definitely click modulation. We can look at sort of more traditional analyses based on just doing what we call click-triggered averages and see things there. We're still working on what is the best decoder for the clicks, because this manifold the activity is unfolding on this manifold over time. And so if you just decode the same way at each moment of time, you won't do as well. And so that's one of the directions that we're taking this.
Or more questions, so
Thank you.
let's catch up during coffee break.